começar? Já estamos atrasados, então. Então vamos começar. Sejam bem-vindos para essa edição do House Symposium. Welcome everybody here. So I will make the introduction in Portuguese, so as it was tradition already, and then we switch to English for the scientific talks. Okay? And the slides are in English because I prepared it a little bit also for my German collaborators, Siegrid Löwel and Fred Wolf, who are visiting from Göttingen. Siegrid was my thesis supervisor, and it's a very nice coincidence that she's here on the 10th edition of the House Symposium. Agora eu vou falar português para vocês introduzir o Instituto para quem não conhece, ou quem está novo aqui, ou os que já conhecem, podem relaxar ainda um pouquinho e se preparar para a ciência que vai vir. Temos, no momento, 18 professores vinculados ao Instituto, o professor Marcos, que voltou depois de um tempo na França. No momento, temos 26 técnicos, e uma particularidade do Instituto do Cérebro, que os nossos técnicos, né, na UFRN em geral, mas particularmente no AIS, muitos dos técnicos, eles já têm um PhD, ou eles já têm um mestrado, ou eles têm uma pós-graduação em andamento, e aos poucos vão ter um PhD, um mestrado. A gente tem sempre alunos de IC, esse número varia, ela é provavelmente bem mais alto, é difícil de atualizar ela. E a mesma coisa sobre os números atuais de alunos de mestrado e de PhD. 25 e 11 são os números do nosso programa nativo, da PG Neuro. Né? Mas temos, além deles, no momento eu vou apresentar números, quantos alunos a gente já graduou no AIS. E porque, em certa forma, no ano que vem, não, o AIS vai fazer 15 anos como unidade acadêmica especializada. Mas as nossas pós-graduações, eles se desenvolveram um pouco como um atraso, e isso se reflete, atraso não, né? O programa nato do Instituto do Cérebro foi criado um pouco depois. Em total, no staff do AIS, a gente tem sempre mais mulheres do que homens. Isso ainda não se reflete nos professores, são os 4 professoras, e isso dá uma percentagem de 22. E temos também 4 estrangeiros entre os 18 professores. Oh! Esse foi muito rápido, mais do que eu queria, desculpa. 26 técnicos, como eu falei. Infelizmente, perdemos a nossa querida Mariana esse ano. Ficamos muito tristes por isso. Mas ela está aqui entre a gente, por isso eu não vou tirar um buraco aqui. Eu queria também mencionar os nossos terceirizados, que são muito importantes para o suporte do AIS, que estão mantendo o Instituto vivo. A gente tem uma porcentagem muito alta na universidade toda, sendo uma unidade acadêmica especializada pequena. Temos relativamente muitos terceirizados, acho que 21 ou 22, porque eles cuidam dos nossos animais. E, de vez em quando, a gente tem até 6 diferentes espécies de animais a serem cuidados. Temos jardineiros e os vigilantes. Faltam 2 vigilantes aqui, porque eles trabalham em turnos. E o nosso, as pessoas da limpeza, muito importante, que estão muito tempo já com a gente, vários dos biotérios também, e Eronildo, que cuida do Instituto tudo. Né? Se estraga algo, Eronildo conserta. As linhas de pesquisa, eles ficaram se inflando nos últimos 15 anos, né? Provavelmente o Raul está olhando, talvez eu estou esquecendo algo que se desenvolveu e a gente ainda não percebeu que essa linha de pesquisa se desenvolveu no Instituto do Cérebro. Mas a gente tem bastante linhagens em volta da pesquisa básica, como vocês sabem, sobre o sistema visual, sistema auditivo, até olfatório, o somatosensorial, sobre comunicação e audição. A gente tem muitas pesquisas que vão até já o comportamento em volta da vocalização e a modificação genética dos circuitos, estudando espécies que são adequadas para estudar isso, os pássaros e os saguis, que estão vocalizando muito. A gente tem muitos modelos em animais sobre doenças, transtornos psiquiátricos, como o autismo, a depressão, a epilepsia, e ainda o tinnitus, que é o zumbido, né, em português também conhecido como zumbido. E estamos indo lá para desenvolver modelos em animais da doença de Alzheimer. Temos neuroendocrinologia, muita pesquisa sobre sono e o efeito de sono também na educação, né? Então, porque nossas pesquisas vão da célula, do canal, né? Da cultura até o circuito e depois o comportamento. E também temos um vínculo muito forte com a bioinformática e com a neurociência computacional, porque o volume de dados necessita isso. Né? Esses são os laboratórios individuais. Eu não vou ler tudo isso, mas eles refletem essa gama de linhas de pesquisa. Eu vou apresentar, e também a variedade das pessoas, né? Isso estava numa outra folha, a formação para fazer neurociência não é categorizada, a gente não tem uma graduação em neurociência. Isso também reflete o que é internacionalmente ainda é assim. Tem poucos lugares que têm realmente uma graduação que liga diretamente para a neurociência. A gente está muito atrelado à graduação da Escola de Ciência e Tecnologia, dando aulas, essas são as redes, na BCT, muitas delas são optativas, das aulas que o Instituto oferece na graduação. Alguns são obrigatórios, porque faz algum tempo, né, a gente está oferecendo um eixo obrigatório ligado à BCT. Mas, em geral, a gente está aula para alunos de todas aquelas disciplinas, e eu deixei isso aqui em inglês para as pessoas que não entendem português podendo ler. E o número de alunos que o AIS está atendendo, ela está aumentando. O Cláudio me sempre deu os números, e eu fiz a contagem de 2025 e fiquei impressionado. A gente está em mais do que 800 alunos na graduação esse ano, nem contando os alunos de medicina, onde Bernadette e eu também estamos dando aula. Então, na verdade, são 920. Yeah? Enfim. Então, a procura na graduação é muito alta. Isso também devido que a gente está no campus agora, com esse prédio bonito, e a gente tem a gente pode receber os alunos aqui e fica mais atrativo simplesmente também para os alunos frequentar o AIS do que se deslocar para um lugar que é um pouco remoto. Né? Mas não só isso. Claro que a procura de neurociência aumentou porque o AIS está mais visível. Isso é interessante para os colegas. A conclusão de teses de mestrado e de PhD desde 2011, desde o Instituto foi criado, e vocês podem ver o total, né, a PhD, que é o nosso programa nato, que surgiu um pouco depois. Essa aqui é a PhD, e essa aqui é a PhD do mestrado, a PhD do doutorado. Como vocês podem ver, em 2011 não se formou um doutor na PhD, né? Demorou um pouquinho, tem um atraso. Os mestrados se formaram um pouco mais cedo. E agora temos aqui muitos, depois da pandemia, principalmente, muitos mestres e PhDs se formando no nosso programa. E antes, né, o que era antes? Os professores que chegaram bem cedo, né, Braulio, Marcos, né, Siddhartha, eles já estavam ligados a outro programa que já existia na UFRN, né, por exemplo, a Psicobio. Né? A Bioinfo chegou também depois. Hoje temos ainda mais programas de pós-graduação, e por isso aqui, por exemplo, aqui está novamente nascendo as teses e dissertações que a gente está fechando em outros programas do que o nosso. mas no total já 110 teses e 112 mestrados. A produção anual do Instituto, em termos de artigos científicos, sempre interessante de ver, varia. No total, desde 2010, a gente já tem 576 artigos. O bibliotecário sempre ajuda nesses contos. Isso dá uma média de 38,4 artigos por ano. Cada ano eu apresento o que era recente. No ano passado, a gente teve outras linhas de pesquisa aqui representado. Agora, a gente tem muito da nossa ciência dos sistemas sensoriais, sono, né, os psicodélicos sempre, e sobre a interação com a respiração com o cérebro. Sempre interessante para o sistema brasileiro, né, é a separação, qual é a qualidade desses artigos. Como vocês podem ver, isso não é que a gente está piorando, isso é porque sempre tem uma coisa que chama histerese, né? A gente faz a contagem, e isso a gente vai ver isso só um pouco depois. E vocês podem ver que os artigos de qualidade A2, né, o A3, eles estão sempre aqui em cima. E também houve uma mudança que o Journal of Neuroscience saiu de ser A1 e virou A2, e por isso também tem uma mudança aqui, que essa curva vermelha está aqui em cima e os outros estão caindo. A visibilidade do Instituto aumenta sempre, né? Isso está mostrando aqui nessas curvas das citações por ano, né? Cada artigo que sai, ele está sendo citado poucos anos depois, e sempre tem um pico em algum momento, né? E isso são as curvas de 24 e 25. Então, nos artigos que saíram agora esse ano, estão sendo citados os artigos que 22, 23 saíram. Mas a visibilidade total, ela aumenta. Importante para o Instituto, não só a graduação, a pós-graduação também é a extensão, né? Isso são todas as nossas ações de extensão que aconteceram em 2025. E eu vou só destacar alguns, né? Um novo aqui, o curso de inverno em electrofisiologia, fez muito sucesso, né? A gente conversou sobre isso com os novos postdocs. A gente tem muitos novos postdocs e pesquisadores mais sênior agora no Instituto, além dos professores. Isso é um desenvolvimento bonito que eu vou mencionar no fim. E uma é a Tour, que é uma ação de extensão onde a gente chama os alunos para conhecer, são alunos do ensino médio, preferencialmente, mas teve procura dos bem pequenos, e aí a Tour veio na escola, veio no núcleo infantil ali em cima para trazer as oficinas do cérebro para lá. E essa Tour, a gente recebendo o aluno do ensino médio aqui, ela atendeu por volta de 300 alunos esse ano, mas já teve anos maiores, e a maioria dos técnicos de pesquisa estão participando nessa Tour, preparam aqui bem bonito os posters, os banners, as oficinas dentro do Instituto, mostras de microscópio dos laboratórios multiusuários, para despertar o interesse pela neurociência e divulgar o Instituto. O Laberto, acho que tem uma outra de Laberto, conversei com o Cláudio, né, que o organizador do House Symposium, na verdade, o professor Cláudio Queiroz, junto com Tarcísio e comigo, mas ele também tem essa ação de extensão bastante interessante, que é o Laberto, o Open Lab, né, que vai nos lugares, são outras cidades esse ano, mas eu não vou saber quais, eu só sei que foram 10 cidades do Rio Grande do Norte, onde essa equipe de pessoas em volta do professor Eduardo, professor Cláudio, foi, e esse ano eles atenderam 2.000 alunos e professores. A Semana do Cérebro, que sempre acontece também atrelado a uma ação internacional de divulgar neurociência. Aqui um exemplo que esse ano a gente teve uma aula no campo, lá na biblioteca em Rocas. last but not least, esse ano foi, apesar das nossas perdas e transtornos assim, em orçamento que sempre temos, foi ano feliz em termos de funding, editais que saíram da FINEP, do CNPq, da CAPES, para conseguir dinheiro. Provavelmente eu esqueci alguns aqui. A gente ganhou uns 5, foi acreditado em alguns 5 ações da FINEP, que ainda não tem todos os frutos. A equipe do professor Traulio, que a Fernanda e o Traulio, eles ganharam o prêmio de inovação FINEP com esse ano. O projeto do professor Tarcísio foi escolhido, foi nomeado, né? O projeto do professor Richardson, e acho que a gente vai ouvir um pouco sobre isso amanhã. Ele ganhou um prêmio de desafio de inovação. E o Instituto faz parte, né, esse ano, ele também foi, foi ainda, né, o dinheiro já está chegando, né, o Instituto Nacional de Ciência e Tecnologia, junto com a Universidade de Pernambuco, sobre neurociência computacional, onde o professor Adriano é o vice-coordenador, o professor Mauro o coordenador. Ganhamos 2 vezes pro Brau, né, que é uma das programas de interação internacional em qual os meus colegas de Göttingen poderiam viajar, e eu posso viajar, que é uma interação com a Alemanha, com DAD e a CAPES. 4 redes no CNPq, com brasileiros, cientistas radiados no exterior, e a gente recebeu 5 cientistas, meio desses editais, que ganharam bastante verba, para entrar numa repatriação, para voltar para o Brasil. E assim se fecha o ciclo, 10 anos, House Symposium, bem-vindo aqui. Eu fico feliz de poder agora já fazer a introdução da minha orientadora, que vai ser a primeira keynote speaker. And now we will switch to English. It's a pleasure to introduce Professor Siegrid Löwel, who was my PhD supervisor, and should I say how many years ago? Why not? Kind of 30 years ago, kind of. And it's a pleasure to have her here, especially because the 10th edition of the House Symposium. And the House Symposium we started in when we had the rented buildings in the Nascimento de Castro, and we didn't have our own room to make our first House Symposium. We were in the Hotel Iramar, was the first edition, and then we sometimes the Instituto de Física Internacional borrowed their space to us. The Metrópole Digital already borrowed their space to us, until we finally could make our House Symposium here, and as it is a tradition, so Claudio, you want to say something? No, we just go on. So I want to thank the organizers, but Claudio did really most of the organization, and many students, and also the equipe, the extension of the Institute was helping. And now the first speaker, so Siegrid, she is biologist, zoologist, actually physiologist, neurophysiologist, would say. And she studied biology in the University of Würzburg, which is more southern, but then soon she switched to Frankfurt because she heard about this fancy Max Planck Institute for Brain Research in Frankfurt, where Wolf Singer, Heinz Wessler at that time were directors. And so she did her graduation in biology, and then her master, which at that time in Germany was the diploma thesis, she started already with Wolf Singer in Frankfurt at the Max Planck Institute for Brain Research, and also her thesis. And when she was already a senior postdoc, I met her, and I think I was the first PhD student. So, and then she continued there, very successful research about the postnatal development of ocular dominance columns, and she worked a lot in cats, but she also, I know that she did ferrets also. And then she went to Magdeburg, where she was kind of a guest scientist, or not yet research group leader in Magdeburg. And after a short period of not knowing where to go, like 1 month, but she como a gente fala aqui, fez questão, que eu falo isso porque faz parte da carreira. 1 month not knowing, being unemployed and where to go, she got a research scholarship at the University of San Francisco with Mike Stryker, and switched the animal model, which was a very smart move, because then the postnatal development research also of the visual system went to the rodents, and she switched to mice. And then she came back to Germany on a guest professorship to Magdeburg and then to Jena. And last but not least, she joined the University of Göttingen, when? 2010, to be a full professor in zoology, and also then even later the Computational Institute of the Biology of Dynamical Networks was founded, where Fred was the founding director, and Siegrid as well, you together. And now she's actually attached to these 2 affiliations. So we are very happy to welcome you here, Siegrid.
So hello, everybody. I really feel honored to be here today on this day and can celebrate with you the 10th symposium of this new and really very beautiful institute. And I must say I'm totally proud of what Kerstin, with the help of you all, has managed to get going here. And I'm very much looking forward to hearing all your scientific talks, and I'm looking forward to lively discussions afterwards. Now, what is my lab interested in? On the I'm the head of the Department of Systems Neuroscience in Göttingen University, and I just put here the webpage if you are interested a little bit more detail. Now, my lab is interested in plasticity of neuronal networks in mammalian brains during learning, during aging, and after lesions. And as Kerstin has elaborated on, initially I started with cats and ferrets, but then I thought either I have to go to monkey or mice, and decided to go to mice because I really admire people doing the monkey work. It's so time-consuming, so challenging. So thank you to all the people who do this work. For me, it seemed better to work with rodents, and I did this, and I learned a fancy new imaging technique in Max Stryker's lab in San Francisco, so I was very happy that he offered me this job. So we work with mice, and I'm still in a visual system, and mice, as we do have a monocular in a binocular part, and I just mentioned this because it's very important, because we work on plasticity of the changes of the UFRNs and their activities. Now, my lab uses various techniques from all kinds of vision tests, including now the latest addition is prey capture, where mice are chasing crickets, and I also will tell you something about this because that's the first really behaviorally relevant vision test that we use. The others were more lab versions. Then we do all kinds of imaging techniques, intrinsic signal imaging, 2-photon calcium imaging in the awake brain, 2-photon spine imaging in the awake brain, and electrophysiology in awake trained rodents. And the two major questions I'd like to talk about today and hopefully answer somehow is, what is the influence of environmental conditions on brain plasticity? That sometimes get a little bit ignored when people raise their rodents just in tiny standard cages. They are not really an appropriate environment for rodents. And the second one is, what molecules control plastic phases in brain development? Why is it interesting to talk about brain development? Well, things can go wrong. And then it would be good if you know why certain critical phases in early in development close, so that we maybe can reopen them for therapeutic purposes. You probably all have seen kids like this. They have one patched eye, and this usually is, the reason is that they have one eye with bad visual acuity, the lazy eye, and then the good eye is patched so that the bad eye is forced to get more visual experience. And already Thorsten Wiesel and David Löwel many decades ago thought about an animal model system to figure out what's going wrong in the brain, in the visual cortex, if there's some problem with vision. And they designed an experimental paradigm, monocular deprivation, where an eye is closed that is meant to simulate cataract or a cloudy lens. And then they did their famous experiments. You probably have all heard about them. In cats, so if you record from visual cortex in cats with electrodes, you see that if you plot ocular diamonds groups, there are some neurons that only respond to the contralateral eye, some only to the ipsi, but the vast majority, of course, to both eyes, because we need binocular vision, for example, for depth perception. Now, something very interesting happens if you use a kitten, a very young animal, and close an eye for just a couple of days, and then surprisingly, only just a couple of days, visual cortex gets activated only by the experienced eye, and no longer by the open eye. And if you do this long enough, this can be irreversible. So the open eye gets better in activating visual cortical neurons. The closed eye loses its influence on the V1 neurons, so primary visual cortical neurons. Now, if you do the very same experiments in adult cats, basically nothing happens. So there's not such a huge change. Neurons still can be activated by the eye that was deprived for a couple of days, but not so in the kittens. Now, I think it's better for the contrast of the slides if you do not illuminate, because we are visual scientists, we want to have good contrast in our images. Okay, so what do we learn from this experiment? There is a critical period for inducing these changes. And we know early in development there is something special with these critical periods. We all know that, for example, second language learning is much easier when we are kids compared to when we are adults. So it's amazing how good cats can speak Portuguese. And also with the eye, there's an early phase where modifications in the sensory input, it's not only vision, can dramatically affect what you can do later on. And that's why we are so interested in what are the rules underlying this closure of these critical periods. Because if we know what's going on, then, as I said, we can use this for therapeutic interventions. Now, what's the state of the art these days in mouse visual cortex research? So how is the timing of sensitive phases regulated here? If we plot brain plasticity versus age, we have initially this high plasticity, and then the high plasticity declines. Now, this is a very, 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 very simplified scheme of the neurons in the visual cortex, or in the cortex. We have 80% excitatory neurons and 20% inhibitory neurons, and I not even plotted all of them. They come in different flavors with lots of interactions. We, and with we, I mean my collaborator Oliver Schlüter and I, believe that, or first, the prevailing hypothesis is that early critical periods close because inhibition gets higher and so-called plasticity breaks are expressed. We thoroughly believe, and we have data, that this is not the entire story, but what is very important are interactions between these 80% excitatory neurons, which are the majority in the brain. And our new hypothesis is that early critical periods close when so-called AMPA-silent synapses do mature. And the maturation of AMPA-silent synapses is, for example, regulated with two postsynaptic density proteins, 95 and 93, that are present in the postsynapse. They are postsynaptic signaling molecules of excitatory synapses that you can have on excitatory and on inhibitory neurons. Okay, now what are amper-silent synapses? Early on, when synapses are born, in the postsynapse, you only have NMDA receptors. You probably know that glutamate is our major excitatory neurotransmitter in the brain, and there are two glutamate receptors, AMPA and NMDA. Initially, only the NMDA receptors are present, and you also know about the activation conditions of NMDA receptors. They are at normal membrane resting potentials blocked by the magnesium block, so they need strong correlated activity in order to be activated. So they are amper-silent, they don't have AMPA receptors. If there's strong correlated activity, because usually you know there's more than one synapse on a postsynaptic neuron, then AMPA receptors can get integrated into the postsynapse, and for this to happen, you need PSD95, for example. Now, so you need the balance of these two molecules in order for a normal maturation to happen. And if you knock out the molecules, all kinds of things can happen, which I will show you now. Now, if you knock out PSD95 in the visual cortex, and there is an electrophysiological measure with patch clamp recordings, my collaborator's lab was done. If you knock out PSD95, then, or first I should say what happens in wild type. If you plot silent synapses versus postnatal days, there's a steep decline. So when the eyes open, then AMPA receptors get incorporated in the postsynapse, and the synapse functions normally. But if you knock out PSD95, there's always this 50% share of AMPA-silent synapses, meaning that you have 50% of the synapses do not depend on PSD95, like are a backbone, not depending on experience and activity conditions, and there's 50% that do depend on, obviously, probably visual experience. Now, what consequence does this high number of silent synapses have for plasticity? And our workhorse usually is intrinsic signal optical imaging, because within minutes you know whether a certain area does show plastic changes or not. So we use, if you use dark red light, and then active regions absorb more of the red light and appear black in images that you take with a sensitive camera from the visual cortex. So it's like a higher resolution version of fMRI. You have a spatial resolution of maybe 50 microns, 50 micrometers. Okay, so if we do this in the mice, and that's the technique that was developed by Valery Kalatsky in Mike Stryker's lab, you see that in rodents, if you stimulate with the IPSI eye or the contra eye dominates input to primary visual cortex. And then we compute an index, contra minus IPSI divided by contra plus IPSI, so we get a histogram that this is positive, so around 0.24, and we color code the ocular dominance map, and orange means the contra eye dominates input to V1. Now, if we close this contra eye early in development for a couple of days, then the two eyes activate V1 more equally strong. We get a leftward shift in this histogram, and in the ocular dominance map, we have cold colors. And honestly, within minutes, you can do this experiment and see whether you have these changes. And then, of course, you can do all kinds of experiment, knock out molecules, change the racing conditions, or whatever you may be interested in. And then you have an in vivo model for plasticity, like an in vivo LTP model, where you can have a screening tool for looking at stuff. Okay, and what is interesting in the context of my talk is that during the critical period for ocular dominance plasticity in mouse visual cortex, so when the animals are up to 4, 5, 6 weeks old, then if you close an eye, what happens is that V1 cannot be anymore as strongly be activated through the deprived eye. So there's a reduction of the V1 activation through the deprived eye, which is, of course, for the experiment reopened so that you can test its input. However, if you do it later on in 3-month-old mice, then you have an increase in open eye V1 activation, which is later. So and people think that this may have something to do with more LTD, long-term depression, happening earlier on, where this is more potentiated synapses. Okay, now, summarize what we have found, because this is all published. PSD95 controls synapse maturation and the timing of critical periods in early development. What is the data? We used wild-type animals of 250 that no longer show this kind of plasticity after 7 days of monocular deprivation. But adult PSD95 knockout mice show this juvenile type of ocular dominance shift. So the adult PSD95 knockout mice behave in this paradigm like 4-week-old animals. Now so V1 activity changes in adult knockout mice are mediated by a reduction of the deprived eye responses in V1 as in juvenile wild type. Just briefly, this does not depend on changes in inhibitory tone. Actually, the inhibitory tone of the adult knockout mice is as high as in wild type adults. So the idea of some researchers that the critical period is closed if you have high inhibition cannot be true, because we can get juvenile plasticity in the presence of high inhibition. As simple as this. And then, to really show that something depends on your maybe favorite molecule, knockout mice are not the ideal solution because you can have all kinds of compensatory changes. So really the proof of concept is that you let a wild-type mouse grow up until it does not show this plasticity anymore, and then you just knock down the molecule of interest, PSD95, just in the area, primary visual cortex, and then we could restore plasticity, like in the juveniles. So we also let GFP express so that we exactly knew that we knocked down PSD95 just in the primary visual cortex, and we did a control knockdown in the animals with the control knockdown, no plasticity in those with the real knockdown, again, juvenile plasticity. And we couldn't only restore juvenile plasticity with imaging, we could also restore juvenile numbers of silent synapses. So it means that you need the PSD95 molecule not only to have initial maturation of these silent synapses, but also to maintain the juvenile synaptic or the adult state, because if you later on knock it out, it reverts to the juvenile state, which is very interesting. And as I already alluded to, there's another molecule, PSD93, which when people studied hippocampus was thought to do exactly the same, more or less. But here in visual cortex, it seemed to do the opposite. So if you knock out PSD93 and look at the percent of silent synapses, they mature faster. So already in the middle of the critical period, or you have nearly no more silent synapses. So we looked at, again, ocular dominance plasticity. So this is the wild type data after monocular deprivation. Of course, wild type mice shift. Very young critical period, PSD93 knockout mouse do show plasticity, but already 3 days later, they don't show plasticity anymore. And we could also phenocopy this with knocking down PSD93, which is again the final proof that it's really this molecule. And very importantly, so ocular dominance plasticity is already absent in late critical period, PSD93 knockout or knockdown mice. And very importantly, this persists in the presence of when you give the mice more enrichment. I mean, as I will also tell you a little bit later, standard cages or animals raised in standard cages are really not normal animals because they are severely deprived. So personally, I only believe phenotypes that you can find when the animals are at least a little bit enriched with a running wheel or have some more enrichments, and I will talk later about it. So they really have this phenotype. Okay, so the idea is then that PSD93 plays an important role preventing precocious synaptic maturation to allow an experience-dependent circuit optimization to happen at all. So you need the PSD95 in order to close what you've learned in this early phase, and the PSD93 to prevent that it happens too early. So you have a balance to ensure proper maturation. Now, what do we have until now? We have plasticity plotted versus age. Without PSD95, they always stay at the juvenile state with respect to ocular dominance plasticity. In 93, they mature too early. So adult PSD95 knockout mice display functional hallmarks of juvenile plasticity. Now the next question was, do the synapses also show juvenile features, morphological structural features? Now, what do you have to do in order to look at single synapses? Of course, you have to do two-photon spine imaging. So what we did is, and because anesthesia, of course, modifies with inhibition in the brain, and inhibitory circuits are somehow involved in plasticity, we thought we cannot do it in anesthetized mice, so this repeated imaging of the same dendritic spines in the visual cortex, we really should do it in awake-trained mice. And what has been known before? So in juvenile mice, if you look at dendrites repeatedly and induce a monocular deprivation, then there is spine loss. In adult mice, there's no spine loss happening. So our prediction was that PSC95 knockout mice should show spine loss. Adult PSC95 knockout mice should show spine loss after monocular deprivation, something that has not been observed in any other adult mouse before. And this was exactly what happened. So, yeah. And for this to do, it was quite complicated because we couldn't just take a mouse line where layer 2-3 neurons are labeled, because we, in addition, wanted to knock down the molecule. So we had to do in utero electroporation to electroporate into the pups while the mom is pregnant, so express knockdown of PSC95 and EGFP, have the mice born that at P45, a chronic imaging chamber was implanted. This looks like this. And then we train the mice to be happy under the two-photon microscope and head fixed. That's quite challenging to do. And then my very talented former PhD student Rashad then did intrinsic signal imaging to be sure that we are looking at a binocular visual cortex. So this is a retinotopic map, and this is how it looks like through the chronic imaging window. So we localize binocular V1, and we have sufficient GFP expression. Now we can look at the dendritic spines in the awake brain repeatedly over days while we manipulate the visual experience and can see whether spines appear or go away. And in fact, it worked as we hypothesized. In the adult PSC95 deficient neurons, we find a spine loss after monocular deprivation. So this is just a zoom in. We imaged the spines over some baseline days, and of course there are some new spines and then they go away, and there's lots of dynamics going on. And if you would just look at slices of the brains, you won't see a difference because there's lots of dynamics. You only see the difference between wild type and knockouts if you look in vivo and see the ongoing changes. Just looking at sections of wild type and knockout mice didn't yield any difference. And this is just to give you an idea of how this looks like. So this is a dendritic segment of a wild type in the initial phase without visual particular deprivation. And there's, of course, always some spines that are lost and some that are added. And this is now in the adult wild types, nothing changes if you close one eye because they are beyond the phase that things happen fast. However, in the PSC95 knockouts, there were also some appearing and lost spines, but then after deprivation, the elimination ratio increased, and you have now many, many more of these red areas. And if we should do the quantification here, elimination ratio, it was doubling in the knockouts and nothing more compared to the normal visual experience in the wild type. So a very, very strong effect. And if you compare the amount of spine loss that we found in the adult knockouts, it was even higher than what other people have observed in juvenile wild type mice. And it was a very clear result. We got a doubling of the spine loss like in juvenile wild type mouse brains. So, and something that was not observed in any other mouse model so far, showing that the adult PSC95 knockout and knockdown neurons display both functional and morphological hallmarks of critical periplasticity. Maybe to mention, we didn't do this experiment just in the knockouts, we also did it in knockdown neurons. That means we had single knockdown neurons in a background, in an ocean of wild type neurons. And the spines on these single knockdown neurons behaved exactly like in the knockouts. So this is cell autonomous. Even if they are surrounded by wild-type neurons, if you have a knockdown neuron after deprivation, there's spine loss on this. Okay, so showing that silent synapse maturation really closes critical period, because also this morphological hallmark does not happen if you knock out the molecule. Now, coming back to the role of critical periods. The role of critical periods is, of course, not that some weird researchers close an eye and look what's going on, because that's not natural. The normal role of critical periods, for example, is experience-dependent maturation of receptive field parameters and development of binocularity. We have to learn. I mean, our brain doesn't have a clue. It cannot be genetically pre-specified exactly how big the eyeballs are, what the distance of your eyes is. In order to get well-tuned binocular neurons, you have to sort it out in an experience-dependent way. So that's one major issue why we have early phases that have to optimize our visual system. And there was a very interesting and influential paper by a colleague of mine, J.C. Kang, in 2010, showing that if you record from single neurons before and during the critical period in the visual cortex, they can have different orientation preferences through the left and right eye. And only when the critical period is closed and there was enough binocular vision, then there's binocular matching of the orientation. So if you close an eye or close both eyes, this doesn't happen. So you need the visual experience in order to that binocular neurons are matched in their orientation preference. So our hypothesis was that maybe our mice can have a problem in this feature because they cannot properly close their critical period. So what was the other thing? We need to record from awake, knockout and wild-type mice, and look at the tuning properties, and we should look at binocular vision and binocular properties with both vision tests and awake calcium imaging. And this is the data I will show now. But first, there was one paper in the literature by Michela Fagiolini from 2003 already, where she observed in younger PC95 knockout mice that they have fewer orientation-preferring neurons, here, wild type compared to knockout. And then I thought, okay, if you have fewer neurons that are tuned for orientation, what effect could this have on vision? And so I had the idea, maybe they are worse in orientation discrimination. So we used the visual water task, which was developed by Glenn Pruski, which is a water tank where mice are trained to swim towards a particular stimulus. And half of our animals was trained to swim towards horizontal gratings, the other to vertical gratings. And then after they understood the task, and their reward was that there's a hidden platform that they cannot see. That's how they see it, so that they can stop swimming. So they are released in the pool and they have to swim. Now, I want to show you a video. I hope it works. So this is a wild type trained to swim towards horizontal, well made, and then it can climb on the platform. This is a wild type trained to swim to vertical, correctly made. Now it gets more complicated. Wild type trained to horizontal, correct. Now I think we have a knockout trained to vertical. Yeah, okay, you made it. But if you make it more complicated and lower the distance, so it should swim to vertical, but it did not do. And if you quantify all the data, there's a huge difference in the orientation angle that you need for the knockout mice to swim properly. So they are much worse in orientation discrimination, but they are very similar compared to wild types in visual acuity. Now you may wonder, okay, so orientation discrimination is significantly reduced in the knockout mice. Then you say, okay, why should I care whether mice are worse in orientation discrimination? What does this have to do with me? Interestingly, is there any relevance for what is happening in the human brain? And interestingly, yes, PC95 is associated with a specific autism spectrum disorder-related syndrome, Williams syndrome. And so authors have checked unaffected individuals and those with Williams syndrome and looked at discriminating orientations at various ages. And what is visible is that people get better with age. But not so Williams syndrome patients. The Williams syndrome patients stay at the early life worse threshold. So the developmental improvement of orientation discrimination is halted in the patients at an immature childhood level. And this deficit sounds very similar to what we found in the knockout mice. Now, does a loss of this molecule result in compromised binocular integration? So the idea was, okay, they are worse in orientation discrimination. Maybe they have a problem in binocular matching of orientations. Maybe they always get a non-matching input to the other eye and are somehow confused. So do they get better if we close one eye? So we closed one eye, and yes, they got better. So wild-type mice in orientation discrimination get worse if they can look just with one eye. The knockout mice get better. Now they are indistinguishable from monocular vision in wild-type mice. So they get much better. So obviously they always get a confusing input that somehow couldn't be matched during development. But, of course, orientation discrimination is not a very behaviorally relevant test, so we thought we need to have a very behaviorally relevant one. Oops, sorry. And this is prey capture. So it was developed a couple of years ago in the lab of Chris Neal. So we thought we need to test our mice in this setup to really see whether in real life conditions, in a behavioral relevant test, they are worse and maybe get better with one eye. And we were lucky and fortunate that Chris Neal and Chen Hui helped us to get the setup constructed in our institute. So what you do is to expose the mice to the crickets, and then they chase the crickets, and then you use deep lab cut to quantify behavior and can quantify all kinds of parameters like how long it takes, when do they start the approach, etc. But first I should show you that vision is really important for this task because, as you know, there's other senses that mice can use. Thank you very much. So this was already published, that vision is important because if you close an eye, they are much worse. No, vision isn't, this is vision important because in the dark they are much worse. Then binocular vision is important because if you close an eye, they are much worse. So capture time is much longer if you close an eye in the wild types. And the third important parameter here is that binocular coordination is used. You may have heard that rodents can have wild eye movements, but not so during approach. So during approach, there are coordinated eye movements and there's maintenance of neutral vergence. So there's, in fact, binocular coordination. So vision is important. Also, they are very olfactory and use their whiskers a lot. So does a knockout of PSC95 modify prey capture? So we set up this arena, and this was part of the PhD thesis of Shupodip, and then used the deep lab cut for quantification of these behaviors. And now I just show you movies, and before I run the movies, this is wild type with two eyes open, so binocular, wild type monocular, and these are the knockout mice, binocular, knockout monocular. And now you can check. And when they found the cricket, the run starts again. So the wild type binocular already managed to catch the first cricket, while the monocular one is still chasing. Oh, the monocular knockout already managed, while the binocular one is still chasing. Maybe we wait until the binocular knockout finally managed to get the cricket. No, still not. Now it's successful. Okay, and if you quantify, it takes a while until they have a stable capture probability, and then we started our experiments. And if you look here at capture time, wild type binocular, very fast, wild type monocular, slower, and then knockout, 2 eyes, very bad, and they improve dramatically with 1 eye. So very amazing. So capture time is much better with 1 eye, and there were 2 other, and even more, just mentioned those parameters, time to first approach, so when they detect the cricket, and total contact duration, how long it takes until first contact that they really catch the cricket. So, and we did it also in the PSD93 knockouts, because as you've seen, the PSD95 knockouts cannot close their critical period, cannot optimize their circuits. The PSD93 knockouts close the critical period earlier. They have a little bit of experience. They can stabilize their synapses, but much earlier. And we hypothesized that maybe they have a deficit because they have a slight orientation discrimination deficit, but not so here in this task. They are normal. Obviously, for this natural behavior, a little bit of experience is sufficient, so they are not compromised, which is very interesting also. So to conclude this part, PSD95 knockout mice are compromised in the binocular, esologically relevant vision task. They have longer capture times, higher latency to detect and initiate first approach, longer time to contact in contact before capture. So, and they improve surprisingly with monocular vision. So proper silent synapse maturation is essential for developing binocular vision in mice. Now it gets even more important to look at single neurons in the awake brain. What properties do they have? And I have to admit that also we started a while ago, we had lots of troubles with our animal authorities to be allowed to do imaging and electrophysiological recordings in awake head-fixed rodents. So it took a while, and we are not yet finished with all the analysis. And of course, in particular, we wanted also to look at whether there's a deficit in binocular matching of orientation preference. But first of all, lots of people helped us getting the electrophysiology setup working, and for the setup, the animals are on a running disc, and they can lick, and they can walk or not walk while they get visual stimuli, and then we use these channel probes here and do spike sorting. Now, I only want to show you a few of the data because these are only preliminary data. We are not yet finished with all the analysis, but things I'm pretty sure that they are correct. That is, first we looked at direction selectivity, and interestingly, direction selectivity is a bit worse. So the direction selectivity index, meaning the higher the index is, the more selective the neurons are, and it's a bit less in the knockouts, which is interesting, and I tell you why, but there was no difference in orientation selectivity. Maybe because we recorded from awake animals and Mikila did anesthetize her animals. Now, why is this interesting? There are ferret data, I think, that show that while you do not need a lot of visual experience for orientation selectivity tuning, you do need it for direction selectivity tuning. And going along our hypothesis, if they cannot stabilize their synapses, maybe that's why they are worse in direction selectivity. Now, how about binocular responses? Because this is the thing that happens during the critical period, and if they can't close, what are effects? Now, what Masoud and Nick recorded is that the percentage of binocular orientation selective neurons seems to be smaller in the knockouts compared to the wild type. And this is what you have to wire up. I mean, initially you have lots of contralaterally dominated neurons, and then the synapses have to get stabilized on single neurons to make them binocular, and if this process is somehow not working properly, then you would have fewer neurons. And another data point that I found interesting is you have a feature called binocular facilitation or summation. So facilitation means that if you present a stimulus to both eyes, the action potential amount can be higher than if you just add IPSI plus contra responses. And this can happen in the mice, while in the wild type mice, here that's the red curve, that's the bino versus the contra, but it never happens in the knockouts. There's never a stronger binocular response compared to a contralateral response. There was really something with binocular integration seems to be worse. And if you look at very simplified scheme here, summation is much less pronounced in the knockout than in the wild types. And now, the data I'm most proud of, because we got a matching result both from the two-photon awake calcium imaging and from the electrophysiology, and this is about the binocular matching of orientations. And in fact, this does not happen as much in the knockouts. So if we look at the delta orientation for the wild type, as also described in the literature, they are around 20 degrees, but the knockout neurons have much less matched orientation preferences in their binocular neurons. And we found a similar result with electrophysiology and with two-photon imaging. So I'm pretty sure it's valid. Yeah, if we summarize this, so in the wild-type neurons, as shown by other groups, you have then in adults matched orientation preferences, but not so in the PC95 knockouts. Okay, conclusions of this part. We believe that, in fact, interactions of these 80% excitatory neurons are very important for circuit optimization. In a way that PC95 is necessary for the maturation and also for the maintenance of the matured synaptic state, while PC93 prevents a precautious maturation to allow experience-dependent circuit optimization to happen at all. And I've shown you that these knockout and knockdown mice show both functional and structural hallmarks of juvenile plasticity, that they have reduced orientation discrimination ability that improves upon monocular vision, that the same happens with prey capture, that they improve monocular vision, and they have reduced binocular matching of orientation tuning. So our data highlight the importance of silent synapse maturation in the excitatory circuits for closing the critical period for ocular dominance plasticity and for developing experience-dependent receptive field properties and binocular performance. Now, briefly, in a second part, I would like to talk about another topic that's very close to my heart. How do you raise your experimental animals? What is appropriate raising conditions? So, most people raise their mice or rats in these so-called standard cages. This is severely deprived condition. Sometimes people put in a running wheel or some more toys. And I always was very interested in this topic, but believing that whatever you do to your mice will influence what you later on, how their brain develops and what you later on can measure, I waited until I saw a standardized setup once at a meeting in Europe, and then I bought the standardized setups. I put in one slide, I mean, basically, whatever you look at is different in mice raised in these bigger cages compared to mice raised in the standard cages. They are earlier eye-opening, visual system development faster, more maternal care, all kinds of molecules are changed. So basically nothing is similar, so lots of changes that are obviously very important, so that mice that you raise in standard cages will never give you the same result that mice raised in enrichment, and then of course you have to think about what is the more appropriate condition. So this enrichment, which is just less deprivation, this alters the expression of key signaling molecules involved in brain excitability, and it will influence brain function and plasticity. Now the cages that I found are these. And so the animals have two floors, one where they have to navigate through a maze, and then they slide down, and then they go through a one-way door, and then they can run. And they love running. My question to you, how much can a mouse run in distance per day if it has the possibility? 10 kilometers. And think about what you do to this poor animal. I mean, not everybody runs 10 kilometers. If it cannot run as fast, as much. I mean, they are in these cages, they cannot even make 2 steps, and then there's again the wall. Okay, so they are very happy, and we change this maze regularly, and they are all queuing to get in, because otherwise, they are really bored to death in the standard cages. I mean, what can they do? Food and water is there. Why should they elaborate on their senses? And then people investigate sensory systems in these animals. Why should they have elaborated on it? And we also learn that things are pruned away if not needed. If you have everything, why should you elaborate vision, somatosensation, you name it? So I strongly would argue that give them a little bit more exercise and enrichment. So does a stimulating environment prevent the age-dependent decline? That was the basic model in neuroscience that I also used. So we took 130 days mice, and they, according also to the literature, they do not show ocular dominance plasticity anymore. If we take mice that were raised in these big cages, they still show this plasticity. And then I thought, okay, how long can we have this? And how about mice that were initially raised in the standard cages and then transformed to an enrichment cage, and they regained plasticity? And our oldest animal was nearly 3 years old. Day 922 still show plasticity. And also they have more shiny fur. So for me, they are more happy creatures. And together with Oliver, we recorded from slices and found that the difference, or one major difference, is that in visual cortex of adult enriched animals, there's low intracortical inhibition, as low as in the juvenile mice. So what seems to be a huge difference, in addition to many other molecules, is that inhibition is lower. Okay, now, it's not only that they stay more plastic for longer, also the changes that you can induce happen much faster. Usually in 4-week-old mice, or in adult mice, it takes 7 days for any shift to happen. In enriched mice, you only take 2 days, you already have a significant shift. And if you are enriched and young, it only takes 1 day for an ocular dominance shift to happen. So obviously, if you are less deprived and more enriched, then activity-dependent changes happen much faster. And there seems to be some cutoff. I mean, if you're deprived, then changes may happen very, 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 very slow. If you have the possibility to run, navigate mazes, then changes happen much faster. So it's not, so the conclusion, the correct conclusion is not that this enrichment rearing accelerates plasticity. I would say standard cage rearing slowly slows down experience-dependent network changes. Yeah, and interestingly, we find this adult type of plasticity that I have alluded to earlier. Now, I want to show you another paper that, when I read this, really got me into this topic. It's from the lab of Larry Fike, Transgenerational Rescue of a Genetic Defect in Long-Term Potentiation and Memory Formation in Juvenile Enrichment. They knocked out a molecule, and it's not important which it was, and then they did slices and recorded the animals with this knockout no longer showed LTP. Nice, you can publish a paper. But then they gave just 2 weeks of enrichment to these mice from postnatal day 14 to 28. And surprise, surprise, the phenotype is gone. If you enrich the mice just for 2 weeks, the phenotype is no longer there. And not only in the mice, but also in the never enriched pups of these parents. So if you have pups born to enriched parents that just had these 2 weeks of enrichment, the pups also did show LTP, although they were knockouts and on their own never were enriched. So defective LTP normally associated with this knockout was masked in both short-term enriched mice and their non-enriched offspring. And in their abstract, they write, the transgenerational transmission of this effect occurs from the enriched mother to her offspring during embryogenesis. If a similar phenomenon occurs in humans, the effectiveness of one's memory during adolescence can be influenced by environmental stimulation experienced by one's mother during her youth. So, if you want to investigate animals, I think you not only should know exactly the life conditions of your mouse or a rodent or whatever, but also of the parents of this animal, because it may have an influence of what you can record or not. That got me into this topic, that we should be much more aware of how we raise our animals and what consequences this has. And of course, I see all the problems. Bigger cages need more space, but I have a solution for you. So we thought, okay, very often bachelor students come to my lab. They only have 3 months of time to do a scientific project. So I said, okay, let's try whether this transgenerational thing would also happen with this ocular dominance paradigm, the simple one. So we put parents in enrichment, then had them mate in enrichment, pups born in standard cage, and checked beyond an age where they normally would not show it, and believe it or not, they showed it. So the standard cage animals, of course, did not show plasticity, but the standard cage raised pups born to enriched parents showed it, and there was more effect of the mother compared to the father. So the presence of ocular dominance plasticity is influenced by the life experiences of the parents. So keep it in mind how your animals lived. It may influence your data, and maybe it also explains a lot of diverging results from different labs. I mean, you probably have heard of all these things, that whether the experimenter is male or female can influence the result of your work in rodents, because they are very sensitive to smell, and hormone levels can be registered, so lots of things can have an influence. Now I have 2 more data slides before I'm finished. Connie in my lab, postdoc Connie, she wanted to test individual mice. Now, see whether individual mice run more or less, and what kind of effect this has on plasticity. And in fact, we found that more running causes more plasticity. So she designed this gated running wheel setup. We chipped the mice, and upon entering the setup with the wheel, we knew exactly which mouse it was and could check all kinds of things. And here we just have 5 parameters: distance, time, speed, how often they entered the wheel, etc. And you see a huge variability from no running at all to, in this case, 4 kilometers per day. And then, of course, we correlated if mice that ran more had more plasticity, and this was exactly what we found. So these are the correlations down, and I just want to show you this slow mouse that basically not ran a lot here, did not shift, although it had monocular deprivation. And then we had Speedy Gonzalez, who ran a lot and showed plasticity. So individual mice react very differently. So it's also good to take into account individual mouse behaviors if you look at your experiment and results of plasticity experiments. Now, of course, it's complicated to have this setup, all the big ones. So the last data slide now is from a bachelor thesis, Fine. We tested whether these running discs that are very tiny, they also fit in the standard cages, could have the same plasticity promoting effect as the bigger cage. And basically, they show the same. So mice without the disc at a certain age do not show plasticity after 4 days of deprivation. But those who had the disc, and we put a camera on to see that they really ran, still show plasticity. So the minimum requirement that I think can be established in any animal house without a lot of effort is give them a disc to run. Same, of course, for rats. Then they are much happier. I'm totally convinced. Okay, yeah, restored plasticity. So take-home messages too. A stimulating environment preserved plasticity throughout life by keeping intracortical inhibition at a juvenile level. It restored plasticity in adult animals, so that were initially raised in deprived cages, then put to the more interesting ones. More running, more plasticity. There's huge inter-individual differences in motivation to run, or access, and maybe hierarchy. There's lots of factors one should take into account. Experience-induced changes happen faster in enriched mice, or also in running wheel mice. So less deprived rearing slows down brain aging and accelerates experience-dependent changes in V1 activation. Standard cage rearing leads to a precocious decline of plasticity and slows down network changes. And I think it's a valid question, what can we learn from mice raised in standard cages? And I would like to advocate, give them at least the possibility to run. They will feel much happier and less deprived. So thanks. Now I would like to thank the people who did the work. Those with the red frame are currently in my lab. I have 2 postdocs, Connie and Livia, and 1 PhD student. Of course, I have to thank the funding agencies. Without them, I couldn't have done anything. And I thank you very much for your attention.
Thank you very much, Siegrid, for this very impressive talk with a lot of data and a lot of impressive new insights. I'm sure we have time for some questions before we go to the data blitz.
Well, thank you for this excellent talk. I mean, massive body of results. I mean, regarding the first part, I caught me wondering how specific would be the influence on plasticity of PSD 95, because I mean, you focused on the visual system, but at the same time you have some behavioral results. So I wonder whether is there any specificity for the system, or is this a general effect in other areas of the brain as well that could also influence behavior? So it's as a general.
After we published the first paper, some bird researcher contacted me, Sarah Bottia, because you know, for song learning in birds, there is this particular nucleus that still that they need for the song learning, and here she found very similar results with reduced AMPA receptors. So she never tested silent synapses particularly, but there was a change in the AMPA receptor responses in this songbird brain, and she thought probably it would
it's
work.
An overall effect.
Probably it's a more general phenomenon, okay. But I would be very happy if people would check their favorite system.
Very nice talk, thank you very much. There are some pediatric neonatal conditions where you have like a decrease in magnesium in the CSF and interstitial fluid, right? So would you expect that in that case you would have like a premature maturation of the silent synapses? And yeah, on MDA, right? So if you have lower magnesium, then you have a easier activation of an MDA, and then the maturation of.
I just think about the result of a colleague of mine, and she works a lot in pushing the concept of inhibition, and he has in his lab has shown that a certain maturational level of inhibition is necessary to open critical periods. And what is a real problem in prenatal care is that these babies are given tranquilizers, diazepam, and you know what diazepam does to GABAergic inhibition. So they are given the tranquilizers, and this may too early open their critical periods, because it would push the maturation of the inhibitory circuits in the prenatal brain at a time where you don't want this, because there's not yet the appropriate experience, because the sensory systems are not yet born. And there was a recent publication where they have checked it in babies together with colleagues, that there was really a change in EEG that they could measure. So that's a real issue that I know of. The others we would have to check.
Hello. So I'm trying to understand how the silent synapses here. So how the silent synapses contribute to the plasticity observed with ocular deprivation? Because I understand that having silent synapses gives you the potential to establish new connections, like functional and interesting. But you seem to show that ocular deprivation results in spinal loss and loss of input from the contralateral eye. So it's like this is loss of synaptic connections. So what are the silent synapses doing here?
on the transgenerational experiments, have you tried to do cross-fostering to check if it's really the paternal-maternal behavior that is?
We haven't done this, other labs have done it.
And what is the answer?
Oh, there's more impact of the mother. Ah, the cross-fostering is the other. I mean, it's known that enriched mothers provide more maternal care. This also.
Okay, so it's not related to the molecular composition of the germ cells.
I think the issue is not completely solved. A colleague of mine worked on it.
Hi, very nice talk, thank you. So I actually have a follow-up for those two questions because I was wondering the same way. So PSD95 is necessary for the stabilization of synapses. So I get the point that when you remove it, you're decreasing the stability, and this instability may allow plasticity to happen. So the expectation here is then you should see this action of PSD95 and 93 in different areas of the brain happening in different times in development. Is that the case? I was just wondering because you mentioned the prefrontal cortex and you see the same thing. Is that in a different developmental time point?
And then the very last question, all the enrichment things, is there any behavioral study you did or you observed where it is a qualitative jump which happens, or is it or did you study if there's some kind of linear relationship between the amount of the animal run and the amount of plasticity it gains? Yeah, it's really linearly correlated.
I also have a paper where non-enriched mice, adult ones, after 7 weeks of mononuclear deprivation, they also show a shift. So if you raise them in this deprived condition, any activity-dependent change just takes much longer. Maybe eventually happen, but you can have it in a day or two if they are having running weeks and not good. That's how it is.
Thank you very much. So we all start running now, and the mice as well.
I started taking the bike to work after taking my car to work after I had these results. And the best anti-aging for humans is dancing. You have social enrichment, physical exercise, cognitive stimulation. You have to remember
John likes to hear